So we're back, and I trust that you've done the calculation. I'm about to give you the answer. So look away if you haven't done the calculation. You don't want me to ruin it for you. You should have discovered that it would require about 2 times 10 to the 11th base pairs of DNA to encode 100 million different proteins, 100 million different genes that encodes a protein of this size. Now, so that's an interesting number, 2 times 10 to the 11th. And I don't know if you know this yet, but every biology teacher knows that the human genome, which we sequenced in 2003, the human genome is made up of 3.2 billion base pairs. 3.2 billion is 3.2 times 10 to the 9th base pairs. And you've just calculated that you would need 2 times 10 to the 11th base pairs to encode 100 million different proteins of this size. So you're off by a factor of 100. You need 100 times more DNA than we actually have. So obviously something is wrong with this calculation. And this is one of the things that fascinated me about antibodies when I first learned about these when I was in graduate school many years ago. So the solution to this conundrum was actually the subject of the Nobel Prize in Nobel Prize in Physiology and Medicine way back in 1987. Say way back in 1987. That was before most of you were born, but for some of us that was like almost yesterday. This was new stuff when I was in graduate school. So there's the person who received the Nobel Prize. His name was Susumu Tanagawa. And you need to go to the Nobel Prize website, read about this particular award. And let me try to explain in one PowerPoint slide what the solution to that discrepancy in the calculation was. And what we see right here is our familiar antibody with the various IgG domains, IgG folds. But what they've done in this graphic is they have associated with this protein graphic the gene that encodes it. So here's the variable domain of the heavy chain, right? And what they show is that the gene that encodes this protein, here's the internal end of the protein, the gene that encodes it is over here. And in the germline cell, the immature B cell that hasn't yet begun to make any antibodies, you have a region of that human genome which has several V gene segments, followed by several more D gene segments, followed by several more J gene segments. And as this immature B cell matures to become an antibody producing cell, what has happened is there's a complex recombination system that selects one of, I think, 230 different possible V segments, combines it with one of, I don't know what these numbers are, but maybe one of 30 D genes. And then those two are combined with one of many J genes. So you actually select one of each of these classes of segments, and you build in every individual cell a different variable domain of a heavy chain. Okay? So the magic here is that we may not have 10 to the 11th base pairs of DNA, but we probably have 10 to the 11th B cells. So every B cell that matures from this precursor, immature stem cell, this germline cell, every one of them recombines these segments in a different way to make a different antibody. It's even more complicated than that because every time you join one of these together, there's a mechanism that scrambles the nucleotides right at this joint and again at this joint. So that multiplies even further the number of possible different sequences that make up this part of the variable region. Remember those hypervariable loops that we saw? This is what encodes, this is what you find in those hypervariable loops. It's a mechanism that is specifically focused on these immunoglobulin genes. We don't do this with any other genes in any other cells in our body. But if you are an immature B cell, antibody producing B cell, then you're going to recombine this specific gene so that you can make any one of 100 million different antibody proteins. Truly amazing. So if you want an even clearer explanation of this, there is a video that we'll provide the link on this website that you can go to. The video is called Immunology Wars: A Billion Antibodies. And it's a short little video that does just an outstanding job of sort of animating this recombination process that's involved. So that's another fascinating aspect of antibodies.